Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. My name is Afiana Rohmani. I come from Medical Faculty, Universitas Muhammadiyah, Semarang. Today, I would like to present my research, the title, A Preliminary Study of Sexual Dimorphic of the 12 thoracic vertebra using 3D geometric morphometrics by computed tomography in the Indonesian population. When the skeletal remains was found, immediate questions that come in our mind will be, are they human? Is it male or female? How old was the person at death? But of all these questions, the most important aspects for identification of the skeletal remains is sex estimation. It is an important starting point to develop the biological profile of human remains, and by knowing if it is female or male, it may be able to narrow down the possible matches by 50%. Several studies have been utilizing a variety of bone for determining sex. However, pelvis bone is the most reliable to use of all bones to determine sex. But in some cases with long post-mortem intervals, such as mass disaster, crime victims, and advanced decomposition, the human remains may be missing, broken, severe fragmentation, or commingled missing. Therefore, it is essential to develop alternative methods using other skeletal elements. The vertebrae are frequently found preserved in archaeological skeletal collections due to their weight-bearing function and relative density. They were usually found to be separated instead of as a complete series. The 12 thoracic vertebra is one of the vertebrae which exhibit the highest classification with the best predictor for sex assessment among other vertebrae. The recent advanced morphometric analysis, which quantifies shape characteristics, is known as Geometric Morphometrics, or GM. The GM is a shape analysis which concerns on the development of coordinate-based methods. From the landmark coordinate briefly encode all geometric data to be analyzed and visualized. Using a combination of multivariate statistical method for shape analysis, this method quantifies variations in shape anatomical objects using the Cartesian coordinate anatomical landmarks. A total of 27 landmarks placed on the digitized 3D CT scan image of T12 vertebra using Stratovan checkpoint software. So this is the description of the 27th landmark placed on the T12 vertebra. This is a preliminary study using a total of 110 samples of CT image abdomen from the radiology department in the Muhammadiyah Hospital Semarang, consists of 50 males and 60 females with age of 20 to 65 years old. The skeletal fracture, skeletal disorders, degenerative bones, which may alter the morphology of the vertebra, were excluded. By the Stratovan checkpoint software, we placed the landmarks on the T12 vertebra. The landmarks were digitized in the same sequence and oriented consistently to ensure it was repeatable and homologous for all samples. By landmarking, the coordinate configuration landmarks were obtained and saved in the notepad format. And then the coordinate configuration landmarks were imported in the MorphoJ for geometric morphometric analysis. By the MorphoJ software, the new configuration landmarks were achieved like this. The blue dots represent the mean landmark position, the small dots represent the landmark positions for individual configurations in the sample. The GM analyses were generated by the MorphoJ. They are Principal Component Analysis, or PCA, and Discriminant Function Analysis, or DFA. The PCA is to identify shape variation among individuals. PCA is done to simplify the landmark configurations and replace the coordinates with the principal components, or the PCs. Since the PCA is to reduce or simplify a large set of variables to a few dimensions, which represent most of the variation in the data, So in this study, we produced the PC scores, which are the projections of the shapes onto the low-dimensional variable. In this study, there are 74 PCs, meaning there are 74 new variables affecting the shape of T12 vertebra. This is the graph showing the amount of variance in the shape of the T12 vertebra. The PC1 account for the highest variance with 21.52% variation among all shape variation, and the PC2 as the second highest with 9.07% among all shape variation. Here we concern on the PC1 and PC2 for exploring the T12 vertebra shape variation, since they account the highest variance. Here is the scatter plots of PC shape variables extracted from the PCA morphoJ in the PC1 and PC2 axes. It is the polygonal curve showing the means of the classifier group, which is the sex group. We can see slightly here that the sex differences mostly shown from the PC1. We can see that the females group, or the red dots, were distributed more towards the positive scores, while the males group, the blue dots, were distributed towards the negative scores. By importing the PC scores from the MorphoJ, we can visualize the T12 vertebra according to the PC scores using the digitized software. The shape changes may explain on the extreme positive and negative scores of each PCs. Discriminant function analysis, or DFA, with cross-validation was performed to analyze whether sex can be correctly predicted with high accuracy. Using MorphoJ, we ran DFA to test how well the T12 vertebra shape may predict whether it is male or female, and the result of the cross-validation test was 60.0% classification accuracy. The t-test was done to evaluate how well the difference in T12 vertebra sizes is between males and females. By the t-test, centroid size of the T12 vertebra showed a significant difference between sexes, with males having higher value than females, with p-score 0.00. For conclusion, geometric morphometrics showed significant variation in size and shape of the T12 vertebra among group different. The result can be used for database collection for the Indonesian population. This project derived an extensive population database derived from the multiple landmarks from CT scan of the T12 vertebra, using GM that will aid in the future victim identification among Indonesians. Thank you very much.